FASTA is a heuristic approach, so as compared to dynamic programming, it is more efficient and quick. And it was written by Lipman and Pearson in 1985. So it can perform optimized searches for local alignment. So now we take two sequences and we are interested in their local alignments, or we take a sequence and we are searching against the whole database. So in this way, we can do those local alignments. We also use substitution matrices here. It allows determination of hundreds of thousands of sequences present together in the database that may be potentially related to your sequence. We call it as a search space as far as that database is concerned, and the sequence you present to this algorithm or this software is called as query. So we take the query and we search in the whole database and we try to get the similar sequences. What is a heuristic approach? So heuristic is any approach that may not come up with the exact solution, but it is real quick and it gives you some optimal or close to optimal solutions, but it is good for immediate goals and it can serve for the purpose. Let's look into some of the properties of this FASTA algorithm. This remember guys, this word is in the beginning it was FASTA. We call it as FASTA, and the same way we get those FASTA sequence which we use in our most of the time. The way we represent our sequences in the databases is FASTA format. So the same people, they came up with that format also. So it's a heuristic algorithm, quick algorithm for shows immediate goals. It takes more execution time than BLAST. BLAST is another fancy algorithm. We'll talk about that in a later on sections. BLAST stands for Basic Local Alignment Search. So it is better for nucleotides than for proteins, and it compares DNA to DNA and protein to protein. So FASTA has some specific parameters, features. So it uses k-tuples. So it takes the small size segments. So it takes your sequence and break that sequence into those short fragments. And for those fragments, it looks into the similarities within those databases. So we call them as k-tuples. Generally 1 or 2 proteins, 4 to 6 for DNA sequence alignments is good for them. So increasing k-tuple value creates less sensitive searches, but we can have more speed. The gap penalties are like this. Gap opening penalty is -12 for proteins and -16 for DNA, and extension penalty is generally -2 for proteins and -4 for DNA. And then it sets the maximum number of reported scores or alignments in an output files. So here is how it works. So we take sequence A and we take sequence B, and within those sequences we try to look into those small chunks or fragments. We align them together, and then we see that along a diagonal line we have most of those similarities. So we have all those segments, we call them runs of identities, some smaller lengths and some larger length fragments. What we do next is we take those fragments and we rescore using some matrices like PAM matrices, and then we keep the top scoring segments. So rest of them, they are trimmed out. Then we join those segments which are retained, which have some scores which is above some threshold, with one another. And then we do some dynamic programming to optimize those alignments. We try to bring them further close, and in this region in the middle, these are the segments which are highly related with one another. So here is how we get a summary of the results which FASTA gives us. So we have sequence length 54, tells about the program, and then we have some expectation upper value, sequence range. We will look into these parameters in a little bit detail later on when we'll talk about those scoring schemes. So this is how your output is displayed. Sequence type tells us that this is a protein, and then there is the version of that software which has been used. Matrix used was BLOSUM 50, number of scores 50, the word size it used was 2, and gap extension penalty was -2, whereas gap open penalty was -10. The output looks like this. So we have those, after seeing those parameters and after running that, we get those outputs. So on the topmost for this sequence, we have this alpha amylase protein at the top. So that has 93.61% similarity, and identity is 85.106. Guys, the difference between identity and similarity is two things are identical, exactly same, and similarity is they are pretty close, similar enough. So we can look into those substitution matrices and we can come up with two amino acids. Looking at those scores, they can be similar or they cannot be similar. So similarities are always higher than identities. The total length of that protein is this one, and the overlap between these two is 47, whereas an e-value, which is a scoring scheme, are calculated. We'll look into that in a later section. So that is 4.7 e minus 13. So these normally those low e's are lower the good. These alignments, they are displayed like this with those scoring colors. So highly similar segments are placed at the top and showing with this red one. So they have up to the highest similarity. The segments are the actual sequence is right here, and these are the sequence which are close to it. So first one, which we have seen in the previous section, so that is right here, and then we have 7, this next one, and so on and so on. So in this way, they are put together, and those colors, they also tell us the strength of those similarities. So more similar red ones, then followed by blue, then followed by these yellow ones. There are different variants of FASTA. You can look into it. So they are different for different purposes. So FASTA can perform optimized searches for local alignments. It uses substitution matrices, and it is a heuristic algorithm.